First off, I want to welcome everybody that is logged on tonight. I do appreciate it. I do see that there's some interested parties, so I'd like to welcome Radwan and Masa. Thank you for taking the time to see what's going on out here. And for the people in the Turks and Caicos and involved with this whole issue, I thought a lot about what you needed to know to do your job out there and what would be most useful for you. And what I realized is that what you guys are facing and what you guys are helping with is critically important out there. This disease is very serious, and Roddy and those guys need a lot of help. And I felt that the best way to arm you or give you the skills to do a good job out there was to give you the investigative approach of thinking about this disease outbreak and methodologies to work with it. So when you go out into the field, I don't want you just sort of following a rote, do as you're told type method. I want you thinking about what you're seeing, interpreting it properly. I hope that questions will come up that then you can direct back at me. Photographs of anything that you don't understand, etc. So this talk will be a little bit longer because of that approach that I'm taking. So I hope you will forgive me that it is a little bit longer and that everybody has a nice cup of coffee ready to go. So what I will be talking about is, I'll give you an overview of this first lecture is an overview of what's now known about this disease outbreak in the Florida Reef Tract, and that information is what you guys need in order to protect your own reefs. So the good thing is that for you guys, now we know a lot more about this disease, and you're way step ahead of what Florida had initially. So that's good. That's going to strengthen your response to protecting your reefs from this disease. So as we know, this is a disease event that has produced significant mortality that it was initially found in Florida region. And when you have a disease outbreak like this that starts to occur, it's occurred in the Miami region, all of a sudden, all these corals were dying. What do you do about that? I mean, as a manager or a biologist or a diver, I mean, how do you even start to study something that you know absolutely nothing about? We see a coral. This is a healthy part. This is the part that's dying, and it looks like it's dying pretty rapidly. So what we need to do is, the path you start down is you start by looking at the spatial patterns of any disease outbreak, this or any others in the future. So basically you need to know, where is the disease occurring, which corals are affected by this disease, and how damaging really is this disease? Is this a cold virus that causes the common cold, or is this a COVID virus that's creating a lot of mortality in your host? So that's how you start down this path of understanding about disease. And then when you have a better idea of what's going on out there in the field, then you can start doing the more focal studies to understand the ecology of this particular disease outbreak. So we looked at things such as confirming that this was an infectious agent, and then trying to figure out what type of pathogen might be causing it. How is it getting transmitted from colony to colony, and from reef to reef, and larger than that, from region to region, so mode of transmission. Do all corals automatically die when they catch this disease? Do they all catch it? Or are there differences among the coral types when they're exposed to this? And then we also want to look for any temporal patterns in occurrence through time in the field. Does it go away in the winter? Is it worse in the summer, or vice versa? What is going on? So all of these answers help us start to try and understand what is happening, what this disease is about. And with that information, that is where we develop the capacity to try and control this outbreak, and all we can really do is reduce mortality from the outbreak, exactly like we're doing as a human species with the COVID infection. Right now, we're trying to reduce human mortality while we're studying it, understanding it, and we're lucky as humans because we can develop a vaccine, get inoculated, and then be protected from that disease, whereas corals do not have that capacity. So we're in a better position than the corals, that's for sure. And then we also want to look and see if there are any environmental cofactors that may be making this disease worse for the corals. Is water quality a problem? Is the disease more severe in areas that are more polluted, etc.? So, as we said, the tissue loss disease outbreak, stony coral tissue loss disease, started around the Miami region, this red region here, around 2014. And the Florida Reef Tract, you can see, extends everywhere where there's green. And the one lucky thing about Florida is they already had monitoring programs in place all along the Florida Reef Tract. So when we see green with no red, we know that the disease wasn't there because there was a monitoring program. So that's lucky. Most other countries, states, areas don't have extensive monitoring like that, and that's a problem. So I do encourage all countries and regions that have associated coral reefs to develop monitoring programs where they can and include disease assessment in that. So we know where it started, and we also know that it has now spread throughout the Florida Reef Tract, and this is just from the AGRA website, and that it has started popping up all over across the Caribbean. So we know where it is, we know when and how it's spreading because we do have people, there are different groups in the water looking out for this disease. So we know where it is, who is it infecting? Well, unfortunately for the corals, this seems to be a pathogen that affects over half of the species found along the reef tract. And we do see differences in morphology. We see more acute lesions that kill faster, subacute lesions that kill slower, and then we see some are multifocal areas of tissue loss. So we're seeing some differences among the coral species just immediately in the field. In terms of how bad is this disease, so we know where, we know who, how bad is it? This is an example. This is Boot Key in Florida. This was a picture taken about a month before the disease hit this reef. If you see brown, that's nice, healthy coral there. And now I'm going to show you a video of this same reef that was taken while I was out there a month or so after the disease hit the reef. And anywhere you see a colony that looks like this, but it's white, that means the disease has just recently sloughed the tissue off, leaving that bare skeleton. So take a look and see how bad this can get. These white ones here, recent dead, dying. Brown is healthy, dead, dead. All of that recent dead within the last month or two at this monitoring site. Just take a look. So it can become very bad, as you just saw. The amount of coral that was lost on that reef in a very short amount of time was scary. So once you know what you're facing, then you want to start down with focal studies to try and understand a little bit more of the ecology of this disease. Now, to understand disease, you have your host, you have your pathogen, and you have the environment in which those two interact. And it's only under certain circumstances the disease actually emerges, especially a disease outbreak. And we call that the triad of disease causation. So just because somebody with a cold sneezes on you and puts rhinoviruses on you, host and pathogen, that does not mean that you're going to catch the cold necessarily. It's not that simple. It depends, were you inside or were you outside with a breeze blowing those viruses away, so you got less of a viral load, and that's why you don't come down with the disease. Is your immune system compromised? Have you been stressed lately or not sleeping a lot? So there's a lot of factors that will determine whether or not disease emerges. And so in order to understand a new disease such as this, where we knew nothing about it, you need to start understanding something about the host, something about the pathogen, and something about the environment that may be affecting that host-pathogen relationship. So that was where I and my group comes into the picture. I'm a disease ecologist. So we were brought into Florida to start working on this disease in 2017. And that is really when the research started. Before that, you had monitoring programs that were following the disease, but nothing else had been done. So this is just a timeline map of the disease outbreak. And I want you to watch, here's the year here, how far and quickly this disease spreads. So everywhere there's red, there's high levels of the disease. So this is 14, 15, 16, 17. So before we even started our research, the disease had already spread through much of the Florida Reef Tract. So the first take-home message I want to give to you guys is rapid response to these disease outbreaks are needed. So the faster you can start doing the research and understanding it, the better your chances of being able to control it. And of course, we know it's now spread throughout the entire past Key West and into the Caribbean. So we started our studies on disease ecology looking at this coral here. This is Montastraea cavernosa, and we were looking at subacute tissue loss. So wasn't killing it too fast, but fast enough. And what we started off, and again, this was 3 years ago when we knew nothing about this disease, the very basics. We started Aquarius studies to confirm infectiousness, look at mode of transmission, and compare how susceptible different species might be. We also used a therapeutic diagnosis method for trying to figure out what the pathogen might need, and then also tagged colonies and followed the disease dynamics through time. So our experiment would be conducted in something like this, where you have water-controlled temperatures and you have everything closed off so that you're assured that you're not spreading the disease or making it worse there. And so this work was conducted in Fort Pierce at the Smithsonian Marine Station there in collaboration with Valerie Paul and many others. Because really, to understand disease, you need a whole team of people with different specialties working on it. So we had some simple transmission experiments. You have an experimental tank. You have the little red X indicates a diseased fragment. You have healthy fragments touching, looking for direct transmission, and in the aquaria looking for waterborne transmission. And then you have a controlled tank, not a healthy coral touching, because corals are aggressive with each other. So you got to make sure that if a lesion says people are waiting in the lobby, did that get taken care of? Any lesions that are formed are from disease, not from one coral fighting the other. And we looked at it in cavernosa and another mounding coral. And the beauty with working with coral is that we can take a single diseased coral and cut it in half and put each half in here, so that we know that these two coral species were exposed to the same pathogens, level of disease activity, etc. So that makes for a nice, clean study. We repeated it looking at cavernosa with a different species, so a Porites species, a finger coral. And what did we find? Just to show you an example, this would be an example of the diseased piece, the touching piece, and the non-touching piece at the start of the experiment. And you would terminate the experiment when you see disease transmission. And so here you can see that the disease has progressed along this initial piece. We've got disease starting here. And in this case, we did not find disease emerging on the non-touching piece. Overall, for the first species, this mounding species with the smaller polyps, all of the coral fragments touching the disease caught it. The ones that were in the aquaria but not touching, there was still 40%, so that's a pretty infective disease. For cavernosa, it was still infective, but less so, and for the finger coral, not at all. Touching or non-touching disease never transmitted to this coral. And the interesting thing about this coral is that where it did touch the Montastria, the mounding coral, you did see the lesion developing, but when you remove contact away from it so that there can't be any coral fighting, what we found is all of those lesions started healing up. And that tells us that was aggression between coral species, not disease, that caused that. So from those studies, we knew that it was infectious, and we knew it could be transmitted both through direct contact and through the water, and that we have differences among the species in susceptibility. Now, as I said, there were different types of lesions, so that was the subacute. So we repeated the exact same thing with more acute lesions that were killing the corals much faster. So for our disease piece, we used this brain coral here that had pretty fast tissue loss. And again, set it up looking at diseased brain coral, touching the mounding coral, and touching the meandrina. Same experiment, and with this, what did we find? For the cavernosa, the mounding, 100% transmission. They all caught the disease. Non-touching, 60%. So this seems to be more infectious than the subacute. For the meandrina, again, all of the fragments caught it, and all of the non-touching fragments caught it. So that is very effective. So we know for disease in the brain corals, it's highly infectious, again, through direct contact in the water column, and both species that were tested were highly susceptible to this disease. Now, this was interesting because if in the field, people have already figured out which are the first corals that start dying from this disease and are most impacted on a reef. And then they look at ones that catch it secondary later on, maybe a month later, and they don't seem to die as quickly as from it. And then there's low susceptible as well. But you see on this, the maize coral was a highly susceptible species, but the mounding coral, or large cup star, was supposed to be intermediate. But when we put these corals touching the disease, they both equally got the disease. So just in terms of innate susceptibility, we weren't seeing differences there. But if you look at the non-touching, then you start to see that indeed that mounding coral was less susceptible than the star coral. And what you then start to look at is you look at the colony morphology. So for the non-touching, you've got to imagine that that pathogen is dropping out of the water column and landing on the coral. And you have to get a high enough infectious dose in order for it to be able to infect that coral. Well, the mounding coral has this smooth, boulder shape, and you can imagine that if stuff landed on there, it was pretty easy for that coral to spit out mucus and push that material off, so thereby avoiding infection from this pathogen in the field. But the star coral you'll see is a much flatter coral, and it has these deep valleys and grooves, whereas if the material landed down in there, it would be much harder for the coral to push it up and slough it off. So, and another take-home message is that the field patterns of infection that you're seeing might not actually translate into innate susceptibility. There may be other factors, such as colony morphology, that may be impacting whether or not a coral gets diseased or not, exposed to this pathogen. So we knew it was infectious. So what is the pathogen? We had already done histology, other people in Florida that had ruled out the larger pathogens, such as fungal infection or cilias. So that basically left it being either a viral infection or a bacterial infection. So what we did is our therapeutic diagnosis, and this work was done by Dr. Blake Ushijima, who is now at University of North Carolina at Wellington. And so what he did is he got a diseased fragment, cut it in half, left one in filtered seawater, and put antibiotics on the other. And the thinking is, if it's an viral infection, then antibiotic treatment will not stop the disease. But if it's a bacterial infection, then it has a good chance of stopping that disease. So he set these up, and here's an example of what he found. Here's the control fragment, day 0, day 4, completely dead. The genetically identical, these were cut in half, exposed to antibiotics, day 0. Ha! Look at that, day 4, that looks just about the same. So indeed, disease was arrested in 12 of the 13 fragments. So this tells us that bacterial agents are definitely involved with this disease. Next, we looked at our tagged colonies to look at dynamics through time. So we tagged 20 colonies off the Fort Lauderdale region, and the interesting thing we found is all 20 were infected. So that's why prevalence, disease prevalence, which is proportion of colonies that are infected, was 100%, of course. But as we went through time, the proportion of colonies that had any disease signs dropped off to very, very low levels. Now, these corals were all these mounding corals, so Montastraea cavernosa. They weren't treated. They were not giving any disease treatment whatsoever. We were just monitoring. We wanted to see what the natural progression of this disease was in the field. And with no treatment whatsoever, we see that corals were actually able to outlive and heal from this. So we see this prevalence declining through time. And accordingly, because the disease was stopped in so many of these corals, the actual colony mortality was quite low. So colony survival through time, and this was a full year you see here, was quite high on this site for this coral species. So what I found is that 3 colonies completely died like this guy. This was initial. This was the next year, dead. 3 colonies healed and stayed healthy through the rest of the year. And then colonies where the disease waxed and waned. 1 colony stayed diseased the entire year, but just died slowly. Some lost it, but got it again. Some lost it, got it again, and lost it. So the colonies were able to differ in their response to this disease. They were on the same reef, the same environment, the same species, yet, and they all caught the disease initially, yet how they responded through time differed a great deal. So you guys are going out there, you're looking at your reefs now, and you're thinking about disease treatment. And so you have to understand what species you're working with, and are any of these species going to heal naturally? So those are all important bits of information as you get out on your reef and start seeing what's going on out there. So we started this by saying, oh my gosh, this is a horrible disease and all these corals are dying. Now you're telling me that these corals are healing and there was kind of low colony mortality. Well, what's the story here? Those are 2 different stories. And this is where you realize that in understanding disease patterns, it is very complicated. It's not as simple as it might be presented. So you got to say, is it just this one species that can fight off that disease better? So is it a species thing that we see differences of tissue loss? Or is there something about the environment in Fort Lauderdale where we studied it that allowed the corals to fight it back better than other environments? Were these caused by 2 different pathogens? Or was there something about the hosts in Fort Lauderdale? Those corals just were tougher than corals in other regions. So you have to start doing your experiments or your studies to examine all these different patterns. So what we did is we took the species question out of the equation and we looked at these same species in our Fort Lauderdale region and then compared it with sites in the lower Florida Keys. And when we did this, what we see is Fort Lauderdale, again, you see corals healing, disease prevalence going down, but the same species in the Keys had the disease the entire time. They either had the disease or they were killed by the disease. And you look at mortality, is same pattern. Low mortality of these species in Fort Lauderdale, but look at what happened in Looe Key. My goodness, those corals definitely died. So it wasn't a species thing, that much we knew. Well, what about pathogen? Perhaps the pathogens were different, or he had evolved for a different virulence on this reef, because bacteria can evolve very quickly. So we went to our aquaria and looked at transmission. And what we hypothesized is if we got a disease fragment from the Keys and touched it to them, that because they killed the corals faster, they would have a higher rate of transmission compared to disease fragments from Fort Lauderdale region, where the disease was not killing corals very quickly. So this was our original hypothesis when we did this. And what we found, here's your day 0. This is when disease emerged. Fort Lauderdale, Keys is in Fort Lauderdale. About almost half of the corals were transmitted. And in the Keys, what we found, oh my goodness, even less transmitted. Now, these weren't statistically significant, meaning basically the rate of transmission was very similar between the Keys and Fort Lauderdale. So just because they were killing the corals faster did not mean that the pathogen had evolved less virulence or that the colonies were less susceptible. So that hypothesis was absolutely not correct. So that makes you think the pathogen may be the same, but what about the potential for secondary infections after a coral has gotten sick? So again, we bring in Blake's work, Ushijima, and we introduce you to this bacteria. This is Vibrio coralliticus. It is already a known pathogen that affects corals in other parts of the world, but it doesn't always. It can be in the environment and not cause disease. But if a particular strain secretes this toxic protein, then that is when disease occurs with this pathogen. So Blake and his group, this was developed actually in Oregon. They basically developed a rapid immunoassay test. So they can put a little bit of that, take a little bit of the tissue sloughing off the diseased coral, and they can put it in these little holes here. It's like a pregnancy test. And if that toxic protein is found in that coral tissue, that will indicate that that Vibrio is in there, and that Vibrio is actively creating disease. And if it is in there, you'll see a second line develop right here, exactly like a pregnancy test. And this is, your test results are available within 10 to 15 minutes. So he got a bunch of different fragments that were brought into the lab for other regions, whether transmission or other projects, and he followed the disease progression through time. So this is day 0, it's clearly disease, and by 16, it hadn't really progressed that much. Here's day 0, clearly disease. By day 4, pretty much dead. And what he found was those that were Vibrio negative did not have that particular bacteria, really survived pretty well. But those that were positive for that bacteria, those are the ones that died quickly. So the Vibrio positive fragments died much faster and had that more acute tissue loss. So maybe this is one of the answers that we were looking for. And he also looked at fragments from Fort Lauderdale and from the Keys, and same thing. If it was the circles, the blue color indicates it was Vibrio negative, and the red indicates it was Vibrio positive. It didn't matter which region it was occurring in. It was that key factor was that if it had that Vibrio, it died much faster. Same thing here. It lost less tissue if it did not have that Vibrio infection. So one of Blake's thinking is that you might have a primary agent that creates this slower tissue loss, and then it is subsequently colonized because it's weakened by this other Vibrio that's creating this acute tissue loss disease. But not all acute lesions tested positive for Vibrio chorolyticus. So although this may be part of the picture, it is not the whole picture at all. So part of it may be secondary infections, but there's still something else going on. So the other thing that we looked at is, was there some natural progression or evolution of this pathogen, or not sure why, but are there changes in these dynamics through time? So what we did is we continued to monitor our tagged colonies. Now in Fort Lauderdale, our site, that disease emerged in that site in 2014. So we didn't even tag that reef until 17, after the disease had passed through. So if there was any evolution of pathogens, or if the most susceptible colonies had already been killed, we wouldn't have even known that. We've only seen it since 17. Our first site in the Keys, it emerged in 18, and that is when we tagged it. So we tagged it shortly after it emerged. We set up a third site in 2019, which also the disease emerged in that same year. So what did we see where the pattern of disease in tagged colony as the years go by? So the temporal changes in disease prevalence, virulence, how quickly it kills the colony, and also lesion morphology. Does that changing as we go through time? So we'll start with Fort Lauderdale, same pattern. Starts, prevalence goes down, we tag some more colonies, some new ones emerge, and again it goes down. For Looe Key, the first year, the disease first emerged here, we see this high prevalence. But look what happened in the second year. Same site, same environment, but the colonies that we tagged in 2019, we see we're able to heal from that disease. 19, that's the first year, analogous to this, and once again we see that those colonies were not able to withstand that disease at all. And for the three lesion types that we're talking about, there's it can emerge as a bleached spot with no tissue loss. We can see subacute or slower tissue loss with these bleached margins, or we see this very subacute or acute with pretty much no bleaching going on. So what did we see at these sites? Well, at Fort Lauderdale, you see when we first tagged them in 2017, we were predominantly at this stage. And remember, disease emerged in that reef in 2014-15, so we missed the initial stage. Now we're here, some with acute tissue loss and a few with just the bleached spots. But look how that shifted. These are the same colonies on the same reef. And we see now most of the colonies just have the bleached spots. Looe Key, the first year, again, a similar pattern. We're seeing a lot of active tissue loss with some subacute, but in the second year, we see that the lesions are shifting. And this is what is now occurring in the colonies there. And at Haslemsley Reef, 2019, again, we see bleached and acute. And we'll have to see how this changes in 2020. So as well as these lesions changing through time, we see this same pattern of mortality. After a while, low mortality, high mortality the first year, lower mortality the second year. Again, a third reef, high mortality on the first year. Consistent patterns. So disease dynamics are definitely changing through time, at least for this coral species. Prevalence starts high, but changes through time. So disease is arrested in tagged colonies as time goes by. Lesions shift from this acute down to subacute, down to this bleached spots. And mortality starts very high, but changes through time. Now, what's important for you guys when you're going out to these reefs and choosing which reefs to treat, not treat, what treatment method are you going to try, you have to keep all of this in mind. What does your lesion look like? When did the disease hit that reef? Because that's going to determine your success rate and how you interpret how successful a treatment is. So in summary, for the ecology of this disease, We know it's infectious. We know it can go through the water column. We know not all species are susceptible. Some are innately immune, and some there are other factors such as colony morphology that make it more susceptible to this. And that the field patterns do not necessarily mean or equal innate immunity. From our field studies, we know that now that the colonies differ in their response to infection, with some dying once they get infected, where others can arrest that disease, and that's with no treatment. So there has to be some genetic component to responding to this disease. We know the disease signs can come and go, and there was no obvious temporal or patterns or seasonality. When we compared Fort Lauderdale versus the Keys, we know now that mortality, lower mortality we found in Fort Lauderdale was not linked to the species, but we did see the disease dynamics were changing through time. Initial infection on a reef, you had high mortality and acute lesions. Subsequent time periods, you have lower mortality and the lesions switch to this bleached lesion. So when we go back to our original question here about how this is all working on disease ecology, we'll start with the host. Well, we know there's some genetic differences in susceptibility among the hosts, but now the next level of research that needs to be done to understand this better, is this due to the coral host, or is it due to this genetic susceptibility, their symbiotic partners? Perhaps their algae that lives in their tissue, their zooxanthellae, or their microbial communities that live on their mucus. We do have to wonder if some reef, the corals start in a healthier state and therefore able to fight this disease off better. That hasn't been looked at yet. And then the host itself. So there are differences in feeding strategy in coral species. Some corals feed on the zooplankton much more than others. And if that disease was being transmitted via zooplankton, then maybe that's why some corals get it faster than others. And we see that perhaps host morphology comes in. Those that are able to slough that stuff falling off from the water column better are more resistant to this disease. We also looked at the pathogen. We know there's a bacterial component and some indication of secondary infections. Is there evolution going on? That's still a question. We know how it's transmitted, but we do wonder about the zooplankton vectors, and we still have to, after we figure out the specific pathogen, can figure out how much of an infectious dose do these corals get before they die. We are just now starting to look at environment, so we really don't know much. We know colony density isn't a major player because a lot of these reefs, in Florida at least, are already low cover reefs, and yet the disease went through there. We didn't see any strong temperature signals, and we do wonder about, again, water quality, because that goes back to affecting health state of the host. So there's still a whole lot of questions left, a whole lot of research left to do, but we know a heck of a lot more than when we started back in 2017, when we all went, I don't know, something's killing the corals. So we're getting there. So with that, I will end this first presentation